Hello and welcome to this Carvin Academy tutorial on AQA A-Level Biology. In this tutorial we'll be looking at enzymes. These are the aims of the tutorial. I'm not going to read and bore you with this, so let's get started. These are the AQA specification points we'll be covering in this tutorial. So let's start by asking the question, what are enzymes? And enzymes are biological catalysts that increase the rate of reaction by lowering its activation energy. And enzymes are water-soluble proteins with a specific tertiary structure. They have a specific shaped active site where complementary shaped substrates can bind. And most enzymes end in the suffix "ase", and their prefix consists of the substrate's name. And some enzymes indicate the type of reaction that they carry out. Some enzymes can also carry out both sides of the reaction, such as hydrolysis and condensation. These enzymes include ATP synthase and lactate dehydrogenase. So let's look at two different enzymatic reactions. Here we start with maltose, which is broken down to 2-alpha glucose by the enzyme maltase. And in this other reaction, pyruvate is broken down to lactate by the enzyme lactate dehydrogenase. You don't need to know this reaction yet, but this will be covered in year 13. Note that on maltose, the enzyme maltase has the prefix malt, indicating that it catalyzes the reaction of maltose to alpha glucose. And note the reaction of lactate dehydrogenase, which is capable of converting pyruvate to lactate and also lactate to pyruvate, releasing a molecule called NAD. So lactate dehydrogenase, the dehydrogenase suggests that it carries out dehydrogenation reactions, and when it goes from lactate to pyruvate, it will dehydrogenate the lactate molecule. Whereas when it's coming from pyruvate to lactate, it catalyzes another molecule called NADH, which you will be covering in year 13. So how does the enzyme function? In order to understand how enzymes function, we need to understand activation energy. To this, let's draw a graph of free energy against time. The reactants at the top and the products at the bottom. And this is symbolic of an exothermic reaction, which most reactions in our body are. And in this reaction, this is a typical curve, and the arrow upwards is the activation energy. You may have seen these diagrams in GCSE chemistry, so this should bring back your memory. So activation energy is the minimum energy molecules need in order to collide with and react. And activation energy is usually provided by heat in chemical reactions outside, but the heat energy required by our body would be so much that it would kill all our cells. So we have enzymes. Now let's have a look at a curve of an enzymatic reaction. So let's have a look at the first part here. And as you can see, the activation energy of an enzymatic reaction is lower, therefore strengthening our point from the previous slide. And the next part is this one here, which shows that the reaction is much quicker as the reaction is ending at an earlier time than the normal reaction. Further to enzyme function, we need to understand how it reduces activation energy. And to be honest, we don't actually know exactly how. However, there are a few hypotheses in how they can work, and one of them is that they put stress on the bonds, and so the substrate molecules can break up. It can also reduce repulsion between two molecules, so they bond easily, and this allows the reaction to occur in smaller steps. So how does this increase the rate? Well, if you have reduced repulsion and put stress on the bonds, you need less energy to break the bonds, which means that for the same energy you can break the bonds more easily, and therefore the reaction will be quicker. Enzymes can function both inside and outside, and they're called intracellular and extracellular enzymes respectively. And extracellular enzymes have the benefit, especially for organisms such as fungi and bacteria, which may lack the mechanisms to carry out the digestion process inside them. So they can release these extracellular enzymes so they can absorb the products via diffusion. And we need to understand how enzymes bind to substrate. And when an enzyme binds to substrates, they form an enzyme substrate complex, a key exam terminology here. And again, we don't exactly know how enzymes bind to the substrate, and two hypotheses were made. Lock and key mechanisms, which are the first earlier hypothesis, and the second one is the induced fit hypothesis. So in the lock and key mechanism, the substrate will only bind to the enzyme's active site if it has a complementary shape, just like how a lock fits into a key. But this means that enzymes can only catalyze one type of reaction, while some can do both. So there's an alternate explanation. This is where the induced hypothesis comes in. So the induced hypothesis, this is a more modern and more widely accepted theory. And the theory suggests that the active site of an enzyme isn't actually complementary before a reaction. But as the substrate binds, the shape of the active site changes so it can form an enzyme substrate complex. There are many factors that can affect the enzyme activity and function. The factors that affect both activity and function are temperature, pH, and inhibitors. And factors that only affect the enzyme's reaction rate, i.e. its activity, are concentration of enzymes and concentration of substrates. Let's have a look at the effect of temperature on enzymes. In order to do that, let's look at a graph of a rate of reaction against temperature. And this is a typical graph of how an enzyme reaction would look. And there are three main points we'll be covering. There's point 1, point 2, and point 3. In the first point, it's clearly visible as the temperature increases, the enzyme activity increases. This is because the substrates and the enzymes gain more kinetic energy and start to move faster. And this increases the frequency of successful collisions, and so more ES complexes can be made. Point 2 is at the optimum temperature for the enzyme, and it's where the highest frequency of collision and the greatest number of ES complexes can be made. And the final point is as the temperature increases above the optimum, enzyme activity drops because they're starting to get denatured. The bonds holding the tertiary structure break, changing the shape of the enzyme's active site, and the substrate can no longer bind, as it's no longer complementary. So little or no ES complexes are made. Next, let's have the look at how pH affects the rate of enzyme activity. But before, we need to understand what pH is. pH is the concentration of hydrogen ions in a solution. Lower the pH, the greater the concentration of hydrogen ions, hence why acids are proton donors. This is a typical graph of an enzymatic reaction against pH. Again, there are only 2 points this time, and the first point is when pH is too high or low, the enzymes become denatured. The hydrogen ions change the position of the bonds holding the tertiary structure, which changes the shape of the active site. The substrate will no longer bind to the active site because it's no longer complementary, so little or no ES complexes are made. The point 2 is the optimum pH, and that point indicates that the maximum amount of ES complexes can be made. So the effect of inhibitors. Inhibitors are molecules that reduce the enzyme activity, and there are two types, competitive and non-competitive inhibitor. In competitive inhibitors, they have a similar shape to substrate molecules, and they compete with them for the enzyme's active site. This animation indicates how the competitive inhibitor tries to compete with a substrate molecule labeled in purple, and as it binds to the substrate can no longer bind into the enzyme's active site, because the competitive inhibitor has bound to the active site before. Note that the competitive inhibitor is not exactly the same shape as the substrate, because if it was, it would be broken down just like a substrate molecule. Non-competitive inhibitors bind to an allosteric site. Allosteric means a site other than the enzyme's active site. And when they bind to this active site, they change the shape of the active site. So what happens is the substrate will be no longer complementary, and so little or almost no ES complexes will be made. And again, this animation indicates how when the change in the shape of the active site means the substrate can no longer bind to the enzyme's active site. So further on to the effects of inhibitors, let's have a look at a graph indicating how they would affect. So this is a normal rate of reaction against substrate concentration. But the competitive inhibitor, this is how it would look. And for the non-competitive inhibitor, this is a typical graph. For the competitive inhibitor, the reaction is actually slower than normal, but it's faster than non-competitive. However, the maximum rate is achieved because it can be overcome by substrate concentration. So if the substrate concentration is greater, much greater than the competitive inhibitor's concentration, the effect can be overcome. For a non-competitive inhibitor, the rate of reaction is very slow, and it also plateaus because the effect can't be reversed, so no more ES complexes can be made. So the effect of enzyme concentration and the substrate concentration on the rate of reaction. There are two main points to this graph labeled here. And for the first point, as the substrate concentration increases, the rate of the reaction increases due to greater frequency of successful collisions. And therefore, more ES complexes can be made. But the rate plateaus because enzymes are the limiting factor here. And no more active sites are available because all of them are occupied by the substrates. So no more enzyme substrate complexes can be made. Now let's have a look at the effect of enzyme concentration on the rate of reaction. A typical graph would look somewhat like this, and there are 2 points to note. Again, the first point is as same as the previous one. As the enzyme concentration increases, the reaction rate increases due to greater frequency of successful collisions. However, the graph then starts to fall because substrates are becoming the limiting factor and the reaction is going into completion. And because there are fewer and fewer sites, active sites are being occupied, fewer and fewer enzyme substrate complexes are being made. So with the end of the tutorial coming up, I'd like you to pause the video now and have a go at these exam style questions from the AQA specification. And these are the answers for those questions. So if you enjoyed this tutorial, please go and have a look at carbonacademy.wordpress.com for more free resources. And thank you for watching this tutorial, and I'll see you in the next one.